like to start by thanking the organisers for inviting me here. I've come to reach one of these meetings, and we get bigger and better each time. I think it's certainly got the most people I've seen in one of these meetings yet. And, it's really great to see so many people who may come in quite a long distance to be able to come and share their experience in this sort of technology. I want to talk a little bit today about what we've been doing in terms of sequencing wheat chromosome arms and how this relates to what we understand about wheat evolution. I'm going to talk a little bit about some chickpea chromosomal genomics that we've been doing. It basically follows up from some technologies that we've developed in wheat and applying it to chickpea to understand genomes. I'm going to talk a little bit about skim genotyping by sequencing, and this is quite different to a lot of genotyping by sequencing methods that you might be familiar with, which are often reduced representation methods. I think reduced representation methods are good and they have their place, but I think skim GBS has got alternative uses, and I'll describe some of those here. I'll describe it briefly for trade association. I'll talk a little bit about gene presence absence variation, because one of the things that we're seeing in genomes more and more is that it's not necessarily SNP variation, which is responsible for phenotype, but quite frequently it's presence absence variation. And the tendency for genes to be present or absent, or to be always there, I think is an important feature of genomes that we're really now beginning to understand, because we're being able to sequence more and more of these genomes. And then I'm going to move on and talk a little bit about some extreme non-model species that we work with, because I think it's useful if you're going to look at plants. You can look at all the different new plants in the world, but if you want to look at some novel phenotypes and some interesting adaptations, we can look at some of the extremes of that biology. So the chromosome sequencing story started back in 2010. I was at a plant animal genome meeting in San Diego, and I was approached by a chap by the name of Jaroslav Bonitzer. And he'd just seen a presentation I'd given on sequencing vascular genomes. We've been doing mostly vascular genome sequencing by then. And that was using a shotgun approach. And he asked me whether it would be feasible to sequence and assemble isolated chromosome arms of wheat. I thought it was crazy. He went on to explain that he could actually isolate reasonable quantities of this DNA at a reasonably high purity. So he persuaded me to do it. I thought, well, it doesn't cost very much, let's give it a go and see what we can do. So we generated some NGS libraries and a little bit of the early and Illumina data technology that was about at the time. And we did a de novo assembly. Just as an overview for people who are not that familiar with wheat, the reason we're doing an isolated chromosome approach in wheat, and the reason why the International Consortium has taken a chromosomal approach to sequencing wheat genome is because it's very big and it's very ugly. It's got 3 genomes, the A, B, and D genomes. Each of these have 7 chromosomes. These are homologous chromosomes. It's massive. It's 17 billion bases. So to try to do a de novo assembly of the whole thing is just too messy. It's very difficult to differentiate between the different genomes. We've got lots of experience of trying to do this in brassicas. We work in brassica natus, which is diploid for taking A and C genomes. And each of the diploid genomes has been triplicated. So we're used to working with fairly complex genomes, but we did appreciate that taking an individual chromosome, or in this case a chromosome arm, would be the best way forward. So we generated some data. The first thing that we did was we mapped it against, this is the rice genome, because we wanted to be able to see if Jaroslav had provided us with some nice pure chromosome arm data, and then what the similarity was with the genome. And as I'm not quite sure with the light how well you can see that, but it maps to one end of chromosome 6 and the other end of chromosome 8. And these are regions which had previously been shown to be synthetic wheat 70S. So this gave us a fair amount of confidence that the DNA that had been provided and the NGS library which had been made is reasonably representative of chromosome 70S. This is mapping of the same data to a synthetic, very zoomed in region, of a synthetic region of brachypodium. And there are 1, 2, 3, 4, 5 genes here. And as you can see with the reads mapping, the reads are mapping predominantly within the exons, the hash regions of the exons. We're not getting very many reads mapping within introns, so there's quite a bit of divergence there. The intergenic regions, there's not really very much mapping. And you can tell quite quickly from looking at this, by comparison with brachypodium, that you have some genes that are conserved in the synthetic region, and some genes that are not conserved. So this gene here in the synthetic region is not in 70S, whereas the other genes are. So without even doing an assembly, we've actually learned quite a lot just from generating this sequence data and mapping it to references. So we're pretty happy with the progress we've made there. So we went ahead and did an assembly, and then we had to ask the question, have we assembled all the genes? And it's not a very easy question to ask because we don't know all the genes that are there. So we did various methods looking at genes which have been bin mapped to the region, various other bits of data that we had there. And we came to the conclusion that we couldn't find any gene that we knew for sure was on 70S. We couldn't, that was absent from our assembly. So we're confident that we'd assembled all or very nearly all of the genes for 70S. And not just the gene regions that you would get from RNA sequencing, we had the introns, we had the promoters, and we had some fairly long contigs which contain multiple genes. We had a little bit of idea for gene order and structure. Also, more than half of the genes we managed to place into synthetic builds based on various other grasses, predominantly brachypodium and rice. So we published this back in 2011, got a print over the Compact Biotechnology Journal, and we were encouraged by this. We were encouraged by the approach. It just seems to be working pretty well. So we went ahead and we sequenced the remaining chromosome arms from group 7. So it's the long and short arms for 7A, 7B, and 7D. And the reason we did this is we wanted to look at root evolution. We wanted to know what the differences were between the 3 different genomes. If they were different enough to be able to differentiate genes from the 3 different genomes, and to basically to look at the evolution and to use it as a tool for gene discovery. So this work was published last year. First investigation shaped the genome of Frederick. And this is a brief description of some of the things that we described in that paper. A little bit of history first, because what we're looking to do is seeing what we can see in the wheat genome, of the 3 wheat genome, and how it relates to wheat evolution. And we know that the tetraploid wheat, the AB tetraploid wheat, formed in the wild 50,000 or more years ago. And it had quite a range, and it was first domesticated in this region around here by the very early Neolithic farmers. So this is some of the first changes of the genome domestication of plants. These Neolithic farmers gradually moved up and took the wheat with them, and eventually they were growing it in this region up here, where it came into contact with the wild B genome wheat. And what was happening now is within the farmer's field, you're getting the ruby B genome wheat growing with the tetraploid wheat. And there was a hybridization event which then caused produced the new hexaploid. So what we're saying is, what all the evidence is for, is that the domestication event was in the tetraploid, and then the hexaploid event happened in the domesticated variety. So we asked whether we could see anything in the genome which would reflect those events. So what happens when 2 genomes come together? They can lose genes, and they often do lose genes, because 2 copies may not be required, and could even be harmful. You also get a lot of methylation and silencing happening, because that's another way that you can prevent these problems with 2 genes interfering with each other. So we asked, can we see differential gene loss between the 3 genomes? I think the A and the B genomes formed first in the wild, and then the D genome came and joined them later. You may expect to see rate of gene loss in the A and the B genomes, which have undergone 2 rounds of polyploidization, compared to genes in the D genome, which have only undergone 1 round of polyploidization when it joined the tetraploid. When we asked, this is what we saw. There's greater gene loss in the A and the B compared to the D. And this is consistent with the pattern of genome evolution. But then we asked another question. We asked, are there differences in the types of genes lost? And this, again, is a bit more of a different question to ask. The reason for this is that conservation of highly networked genes are conserved under neutral selection. The genes that are parts of large networks that interact, the proteins interact with each other and are reliant on one another, there's an evolutionary constraint on them. They do tend to be kept together under neutral selection. However, where there's strong selective pressure, the networks tend to break down because there are other forces that come into play relating to selection of parts of the genome. So we're asking whether we're seeing signs of a conservation of neutral selection or strong selection. So we wanted to have a look at the gene networks within the different genomes. We couldn't do this directly on wheat because the genome was still only part there, and there's a huge challenge to get very good genome networks up and running. There's a lot of genome network analysis being done in Arabidopsis. So we decided to piggyback on Arabidopsis. It's not ideal, but it did seem to work. And where we were confident of an ortholog between wheat and Arabidopsis, we had a look at the gene network. And these are genes which are on 7A. And as you can see, many of them are conserved in the networks, and there are a certain number of genes which are not in gene networks. If you look at the B genome, you see a similar sort of pattern, maybe a few more genes that are not in gene networks. When you look at the D genome, you see there's far, far, far more genes which are not in gene networks. So what did this tell us about the evolution of the wheat genome? Well, you go back to what I was saying earlier about the tetraploid forming about 50,000 years ago. This happened in the wild. It was under neutral selection. There wasn't a great deal of selective pressure when it happened. It probably happened over an extended period. However, when the hexaploid formed, this was in the early farmers' fields. These are where the farmers were presumably planting out the tetraploid that they domesticated, and then they were crossing with the D genome. There was probably a lot of selective pressure for that new hexaploid to become domesticated. They were choosing specific ones that looked like the good ones who had the domestication trait. So you would expect to see strong selective pressure under the domestication scenario compared to the non-domesticated scenario. And this is what we think we're seeing in the gene network. It's a reflection of the early farmers 10,000 years ago selecting particular plants, and that is shaping the three different genomes. So moving on, I've been assembling genomes for a few years now, but I've been doing working with molecular markers a lot longer. We've developed a range of different software for identifying markers. There's quite a bit of bioinformatics in this area. And this is our latest product. We published it in Biology last year, I think, or the year before. This is SGS AutoSNP. It's designed to identify SNP markers by comparing Illumina short read data to a reference. The first version that was published only worked with homozygous material, so it was fine for wheat. We've now established a version for heterozygous material. It's a bit different from a lot of other SNP discovery software, particularly stuff that's been developed for humans and other vertebrates, in that it's working mostly with complex genomes. It's not there to identify all possible SNPs. It's only there to identify SNPs that we can confidently identify. So the accuracy is fairly high, but at the same time we're not capturing everything. Also, it identifies SNPs by comparing all the short read data together with the rest of the short read data. It doesn't identify SNPs by comparing short read data with a reference. The reference is only there to map the reads. So we developed that bit of software. At a similar sort of time, we were part of a consortium in Australia to develop resources, genomic resources, in this case genomic resources for wheat. And what we managed to do as part of this consortium, we managed to sequence 16 Australian elite bread wheat varieties to 10x coverage. That was still pretty expensive now. It was really very expensive then, but we managed to persuade people to pay for that. And we did that because we thought it's a pretty useful resource. I'll explain what we've done with some of that data and applying it to the SNP discovery as well. So we ran all that data, mapped it all to the group 7 chromosome arms, and we identified 4 million SNPs. Most of the SNPs are outside of genes, and as you can see from here, there's a greater SNP density on 7A and 7B than there is on 7B. This has been described before, and there wasn't really a very good explanation for this. We asked quite a few people what could be the reason. And there's quite a few theories that were put forward, but the most convincing one that was come up with appears to be that, again, going back to the farmer's field. The farmers 10,000 years ago were planting the tetraploid, and they were probably growing it with new hexaploid. They didn't do a psychological analysis. They didn't know which was which. They were just choosing the best season time to plant. So presumably quite a long time, these are growing together. However, the farmers wouldn't really be planting the weedy species. So there's probably some weeds around there, but it wouldn't be a huge amount. So what we believe has happened is that there was genetic exchange between the hexaploid and the tetraploid, but not between the hexaploid and the D-genome wild species. And this is what's caused the higher SNP density, the greater amount of variation in the A and the D genomes of the hexaploid compared to the D genome. We've done quite a bit of analysis moving across the syntenic builds for the three different chromosomes, as well as seeing the difference. We just look at the scale of there being far fewer SNPs across the D genome. You're actually seeing quite a bit of variation across as you go through. And one of the questions we asked with this data was, are there any particular genes or classes of genes which are in regions of low SNP density? So the reason we're asking this is that we would expect if there are particular genes which conferred beneficial agronomic traits in these 16 varieties, you would see selection for these, and therefore you would see reduced a number of various haplotypes, and you would see reduced number of SNPs. So when we did the analysis, we identified the genes which are in low SNP density regions, and then did some genotypology analysis. The sort of genes that were coming up appear to be the sort of genes that you may expect to be selected for. So this could be quite useful if breeders are going through and looking for particular genes. If you know that a gene is important for an agronomic trait, say for example because it's important in a different species, you might want to have a look in wheat and see if it's been selected for, see if there's any real diversity, see if there's other varieties that could bring that diversity in. So from a breeding perspective, we believe there's some use in this to be able to put and annotate on the genome database whether the genes are in diverse regions or regions of low SNP diversity. These are the different varieties that were sequenced to identify the SNPs. If you look at a pairwise comparison, you see quite a bit of variation. I think there's almost 1 million SNPs between Yixi and Tara, and there's very few between Tara and Axter. But in all of them, there's at least 100,000 SNPs on group 7, which I think is more than enough SNPs for most purposes. So it's easy enough to draw a tree from this, and if you wanted to start selecting diverse varieties for doing some trait mapping, etc., and wanted to increase the amount of diversity that you had in your population, this sort of analysis allows you to go and choose the ones you wouldn't choose, ones which are most similar, you choose ones which are most diverse. All the wheat work that we do is publicly funded. It's predominantly a hobby. I don't have a huge amount of wheat funds, and I believe it's very important that the data is made freely available as quickly as possible. So we always make our data available pre-publication. A lot of the stuff is presented as being published, apart from the SNP stuff, which I think is in review at the moment. But all the data pre-publication is available on the website to download. Or on the short read archive, etc. I think wheat is too important to crop to have it protected. I think it's important that as soon as the data is generated, it's out there for people to use and get breeders tools and breeders can actually use it to improve it. So moving on from wheat, wheat chromosome sequencing seems to work pretty well. So we started to think about what else we could do with isolated chromosomes, and I was working with the receiver, many people in this room on the project to sequence the Kabuli chickpea genome. At the same time, I spoke to Yaroslav and asked whether it was possible to isolate chromosomes, individual chromosomes of chickpeas based on their size. And he didn't know, but he was happy enough to give it a try. And he managed to isolate all chromosomes individually, apart from chromosomes 4 and 7, which you could isolate as a pair. So what we wanted to do with this data is to map it against the published Kabuli reference genome. Simply ask the question, how good is the genome? Are there any problems with it? And generally the genome looks very good, but there are regions, if you have a look here, regions, for example, where you're mapping chromosome 1 isolated sequence data. So the chromosome 1 reference. However, when you look at other chromosomes, look at chromosome 2, I think that is probably 6, and 3, we're getting reads from these other chromosomes mapping into these gaps. And what this is telling us is that there are a few small errors, just over 40 regions where the assembly has been misplaced. We've got a region which we're saying is a particular chromosome, it's actually a different chromosome. Now, this is making an assembly bad. I think this assembly is particularly good. I think if we did this analysis with Medicargo or any other genome that's out there, then I think we're going to see similar sort of analysis, similar sort of issues. And I think this is a really useful way to be able to go through and validate your assembly, and to be able to say, okay, these regions need to be put to the correct place. One of the advantages of it is the resolution. Because these are reads, short reads mapping to a reference, you've got almost base pair resolution of the region which has been misplaced. So we just basically put that and then put it somewhere else. We asked the question, are there, what was the cause of the error? Was it configs that have been where they've been misassembled? So during the config generation, we've got different regions coming together using chimeric configs. It seems to be a bit of both, and mostly caused by transposable elements, which is what you'd expect. So another question we asked is how similar to the DESI and Kabuli genome. There's a lot of suggestions that DESI and Kabuli should effectively be the same. They've been interbred in various breeding programs. So we've got we asked how to isolate some DESI chromosomes from Kabuli chromosomes. So this is DESI and Kabuli chromosome 8, DESI and Kabuli chromosome 3, Kabuli chromosome 5. And as you can see, actually this mapping is quite useful for validation here. We're not seeing any real differences between DESI and Kabuli. So this supports what was previously described in that DESI and Kabuli should effectively be the same. However, a few months after we published the Kabuli genome, the DESI genome was published. So we downloaded that reference and we had a look and did a comparison. Standard dot-plot comparison of the 8 chromosomes in Kabuli as compared to DESI. And we saw some quite significant differences. What we're saying is chromosome 8 in Kabuli seems to map to chromosome 3 in DESI. They're saying chromosome 8 seems to map to chromosome 7 in Kabuli. There's a lot of differences that we wouldn't expect. So we asked the question, can we use the isolated chromosome data to figure out what's happening here, to figure out if there are major differences. So what we did, we took DESI and Kabuli isolated reads, the 2 chromosomes that we had before, and we mapped them to the DESI reference. And what we could see was that chromosome 5 in the DESI reference looks pretty good. It's fairly clean. We've got both DESI and Kabuli chromosome 5 references matching to it. Chromosome 3, we're getting both chromosome 3 specific reads as well as chromosome 8 specific reads mapping to the chromosome 3 reference. And what this is telling us, we're not getting anything mapping to the chromosome 8 reference. And what this is telling us is that within the DESI reference genome, the chromosome, what they're calling chromosome 3, is actually chromosome 3 and chromosome 8 merged. So there are a few issues. And so we probably do believe that what they're saying there is that chromosome 8 probably is chromosome 7. What perplexes even more is these kind of bluey purple bits here. We see most things are orange, and most things are orange because the chromosomes are not isolated to 100% purity. You get some little bits of chromosomes coming in, and you can see that quite clearly with chromosomes, especially to the next to each other, getting quite similar, a bit more orange compared to the red. These blue bits are where there is no read mapping at all from the isolated chromosome arms. And this really perplexes me. So we got some whole genome data for DESI. I should point out that the DESI chromosomes were isolated from the same variety as the reference. It's not a reference. We've got some whole genome data for DESI, and we mapped it, and we're seeing exactly the same pattern. So what we're concluding from this is that these regions here are not even DESI, they're not even chickpeas. No idea what they are. You have a look at them, there's no genes in there. The GC content is similar to the rest of the chickpea genome. There's no structures that look like it could be genomic DNA. There's no transposons or anything like that in there that look quite similar. So we're a bit perplexed with that. And I should say I have actually presented this to the authors of the DESI genome paper, and we discussed how we can help them improve the assembly. And this is down there. This shows that that you shouldn't really trust any genome assembly. The Kabuli one isn't perfect. Everyone certainly isn't perfect. I think any genome should be taken with a pinch of salt, and there's lots of potential to improve a genome. Now, it's very easy to find faults in these things. I've been married for almost 11 years, and my wife's still finding faults in me. It's very difficult to fix those faults, to come up with ways to fix things. So rather than just going through and saying these are where the problems are, we want to be able to go through and find ways to fix genomes. And one of the ways that we've been doing this for some time is the skim genotyping by sequencing. And I'll describe in a little bit what I mean by skim genotyping by sequencing. But essentially we do lots of SNP calling. We make meta-SNPs for configs. So within a particular config of the genome, we're going to have lots of SNPs. There's very little recombination within a config, so we just call one SNP that represents those regions. We can merge configs where the haplotypes are similar, and I can show you that. We can do some genetic mapping similar to the top-seq approach. We do some comparison of all blocks based on applied clustering approaches. And I'll describe this in a bit more detail. It's really quite interesting. So the skim GBS is different to things like RAD GBS or other types of GBS in that it's a two-stage approach. First stage is we identify all the SNPs. So we essentially run SGS order SNPs and identify all the SNPs between the parents. So we need a reasonable amount of data for parents, maybe about 10x coverage for each parent. Once we've identified the SNP, we then do low coverage skim sequencing of a segregating population. And then we map these regional segregating populations to reference genome, the same reference genome that we call the SNPs from. And we call a genotype where a read covers a previously defined SNP. Once we've done that, we can impute missing data and clean to define haplotype blocks. This is what it looks like graphically. We first identify the SNPs in the parent, and then we take reads from the segregating population. Here we know there's a CA SNP at this position. Here the read says that's an A position. So we know in the individual from this that this genotype is an A, and we just do that across the whole genome. This method has got an advantage in that it's tunable. If you want to be able to call pretty much all the SNPs that are in the population, about a very large number of SNPs, you just dial up the amount of data, you generate a lot more data. If you don't need very high SNP density, if you just want a genotype of a very low SNP density, you just generate a relatively small amount of data, and then you can impute the missing data. So for genome validation, for genome fixing, we need relatively high resolution, we generate a lot of data, and for things like trade association, we can generate a lot less data. This is what essentially looks like. We have haplotype blocks. This is in brassica, this is Capidor, this is Lingu, and what we see in the segregating population, if all goes well, you can see conserved haplotype blocks for this individual or this region has got the Capidor allele, it's got the Lingu allele, and here we see the combination. In practice, it's a little bit messier than that, but this is generally what it looks like. When you start to impute, what you can start to see in these is regions where you have the haplotype being conserved, and then across all individuals, so these are SNPs across the top, very large number of SNPs, these are individuals going down with different random colors. You can see regions of the reference, here and here, for example, where the haplotype breaks and then goes back again. And there could be two possible reasons for this. If the population is different to the reference, it could be that there's variation in the genome structure. If the population is derived from the reference, then what this generally means, in this case, it means that that this region of the reference is being incorrectly placed. So this is similar to the chromosomal genomics approach, where you identify regions which are different. But the advantage of this is that this is essentially a barcode. You can take this pattern and you can ask whereabouts in the genome you've seen this pattern. And from that, you can then place these regions into the correct position. We've been doing this for a while with a few different approaches. So this is based on a clustering approach. We do some initial clustering. This is one of the brassica genomes. We've got 8 chromosomes. And this is just a similarity matrix of the various SNPs against each other. So you have a white line going down the center because all the SNP patterns are identical to themselves. And what we can see clearly is that the SNPs or meta SNPs within regions which are misplaced. So this is what here we initially said from the genetic mapping that this was based on chromosome 3, actually belongs on chromosome 2. You can then apply various different clustering algorithms to start to improve the assembly. You start getting patterns like this. And what you see different patterns. You see greater similarity in haplotype block patterns with the centers, successful combination there. And you get some really quite beautiful pictures. And this tells you a few things, but it tells you mostly that the genome is fixed and is pretty good. I'm not going to talk very much about trait association, but if you think normally about anything you do with skip discovery and GWAS and running GAPIT, you can do with these skips as well. There's a couple of examples of disease resistance in canola, there's that tolerance in chickpea. It's exactly the same method as you would use with rapeseed or Illumina or Illumina. So moving on, I'm going to talk a little bit, I'm going to speed up a little bit. Gene loss. So SNPs are important. We think gene loss is more important than SNPs. So how do we identify gene loss? One of the easiest ways of doing it is to map reads to a genome where reads don't map, the gene is lost. Relatively easy. So this is an example from cabbage. We're taking some cabbage whole genome data. We're mapping against the 2 of 1,000 factorial ratio reference. What we can see, these are conserved genes up here, and these are lost genes down here. We can do it for a whole range of different brassicas, sprouts, cauliflowers. Brassicas are wonderful. They've got very similar genome structure, very diverse morphology. Kohlrabi, it's a wild brassica roll ratio. There's a lot more genes lost in here. And what we're doing with this, we're listing all the brassica genes. We've got all the brassica genes, we're identifying the new brassica genes, which are new assemblies, and we're putting them all together, and we're categorizing them and saying, oh, these genes are essential, these are always conserved, these genes are optimal, these genes presence absence variation. Once you've got this information, you can start to associate the presence absence variation with traits. There's one lot of evidence that we can very easily, especially through that disease resistance, but also for other traits, which we associate presence absence variation with these traits. Also, there's quite a bit of evidence coming out now that the abundance of optimal genes, if you start to get all these optimal genes in the genome, the more of them you have, you tend to get a fitter plant. So there's a potential to actually do crop improvement based on selection for more and more of these optimal genes. This is still very early stages, but I think it's still in the right direction. So, I think I'm 2 minutes. Give me 2 minutes. This is not Somerset in England at the moment. I know they've had a lot of rain there, but not quite this much. This is one of the extreme species that I've been working on. This is seagrass. This is a manatee. This is a sea cow, one of the main feeders of the seagrass. Seagrass is pretty important. It's a grass. It is what it says it is. It's a grass. It's a monocrop that evolved to live on the land, as most crops have done, but then evolved again to move back into the ocean. So the evolutionary path is quite interesting. It's interesting from an environmental point of view. It's the major carbon sink in the ocean. It's a major store of carbon. It produces lots of rhizomes and stores lots of carbon in that. It's a long-term carbon sink. It stays there for thousands and thousands of years and gradually turns into oil. I think what we're doing here is essentially working for the future oil industry. It's also important for fish stocks. It's important from an environmental perspective, from an evolutionary perspective, because it's a plant that's evolved to live with a lot of sodium, high sodium potassium ratio. So if we actually look at this adaptation, this extreme adaptation, and learn from it to apply it to crop plants, it could be quite useful. Now, I did extensive research on seagrass to look to see any other applications of it. And particularly, I found one involving the manatee here. And apparently, this is a real website. Someone's actually looking to make cheese from manatee milk. I'm not quite sure exactly how they're going to do this, but apparently, maybe it is delicious. I haven't tried it yet. We were more interested in gene loss. So we asked a fairly strict question. We had a look at genes that were conserved among various land plants. So these are genes that were conserved in these 4 species. And we asked if these genes were conserved in seagrass. And the ones that we saw that were lost, the genes that are present in the land plants, were lost in seagrass. Some of them are ones that we expected. So we know that the seagrass doesn't produce proper flowers. It does produce pollen, but the petals, it actually has little leaves covering it instead of petals. Genes involved in petal formation are obviously absent. Hasn't got any stomata. Stomata are not particularly useful if you're living underwater because if you have any stomata, you tend to drown. So they don't have stomata, so genes for stomata are lost. One of the things that we hadn't expected, but we probably should have expected, is the genes for ethylene biosynthesis. So the genes for ethylene biosynthesis and ethylene perception are absent. This plant doesn't produce ethylene, it doesn't respond to ethylene. And it actually makes sense when you think about it, because what's the point of having a gaseous hormone underwater? It's not especially useful. So for people who are studying ethylene, especially ethylene response to stress, it could be an interesting plant to work on to see how this plant works in such a stressful environment. A lot of the genes that we're seeing in seagrass have diverged significantly from the land plant versions. And one of the things we're looking at specifically is the genes that are sensitive to the sodium-potassium ratio. Seagrass is very good at pumping sodium out. It's got various mechanisms for excluding sodium. However, within the cytoplasm, the sodium-potassium ratio is similar to a highly stressed wheat plant, and yet the seagrass isn't stressed. So what changes to the biochemistry and the enzymes in seagrass are allowing it to survive. So we're looking at, we're studying the CSIRO, some of those modifications. Conclusion. So we built high-quality reference genome assemblies with this data. We've identified variation between genomes. We associate genome variation with agronomic traits, and we've applied diverse genomic knowledge to improve crops and lots of different crops. Half a minute to thank everybody. This is my group currently. This is people who contributed before from my group. Jackie's group, we wouldn't be able to do anything without her. Yaroslav's group on the wheat and the chickpea. These are other collaborators on chickpea. Many of them are here. And these are the seagrass people. And finally, a quick ad for a meeting. If you're interested in agriculture and climate change and want to go to Amsterdam for some reason in February next year, please do. And I'm happy to take questions if there's time.